Today, we're diving into the world of single-cell RNA sequencing using the Seurat pipeline for 2024. If you're a bioinformatics enthusiast or researcher looking to analyze complex cellular data, this guide is your go-to resource. Single-cell RNA sequencing helps us understand the gene expression profiles of individual cells, revealing insights into cellular functions and diversity. Seurat, a powerful R-based package, simplifies this process. The Seurat pipeline includes 5 key stages: data preprocessing, quality control, normalization, clustering, and visualization. Each stage plays a crucial role in ensuring accurate, meaningful analysis. First, data preprocessing cleans raw sequencing data. Quality control filters cells based on metrics like gene count and mitochondrial content. This ensures you're working with high-quality, reliable data. Normalization adjusts gene expression levels to eliminate technical variations. Seurat's log normalization method ensures consistent data scaling, making cells comparable. Seurat's clustering algorithm groups cells with similar expression profiles. Dimensionality reduction techniques such as UNAP or TSNE help visualize these clusters, revealing patterns within your data. This pipeline offers a comprehensive step-by-step approach to single-cell RNA-seq analysis. To get started with Seurat and uncover deeper insights into your data, visit the full article linked below.